So we finished lecture talking about Barr body formation and the inactivation of X chromosomes. So this is the process that we see in mammals where the female individual has 2 X chromosomes and the male individual typically has 1 X chromosome. And the whole mechanism that kind of has led to this process is a way to manage the gene dosage between male and female individuals. So if you have 2 copies of the X chromosome, you have twice as much gene expression of X-linked genes compared to the males who only have 1 X chromosome. So they can only produce gene expression from that single X chromosome. So dosage compensation is the idea that 1 copy of a gene, or in this case an entire chromosome, is effectively turned off or silenced so that there's an equal amount of gene expression between 2 types of individuals. So again, in this case, female versus male. Okay, so we talked about lionization. So this is the process of X inactivation. Lionization is named for the person who observed this first. We are talking specifically about expression of the XIST gene. This is found on both X chromosomes, so there's 1 allele for XIST here and 1 allele for XIST on the other X chromosome, only 1 allele down here in the males. And at the very beginning of development, post-fertilization, you have weak expression of XIST from both X chromosomes in female individuals. And over time, once you get to the blastocyst stage when differentiation is starting to occur, 1 of the copies of XIST is going to be silenced. So in this diagram, the copy of XIST expressed from the left-hand chromosome is silenced, and it continues to be expressed from the right-hand X chromosome. XIST, shown in pink, is specifically a non-coding RNA gene product that never gets translated into protein. It only functions in its RNA form, and it forms an envelope essentially around this X chromosome. XIST plays an important role related to what we've talked about with histone modification, in that XIST non-coding RNA, so this stands for long non-coding RNA, is going to be recruiting methyltransferases and deacetyltransferases. So previously today we talked about HAT, histone acetyltransferase, and how that protein adds acetyl groups to our histone proteins, those histone tails. Acetyl groups increase gene expression, and so the opposite of that is deacetylation, or taking away acetyl groups, which would have the effect of reducing gene expression. So in this context, long non-coding RNA from the XIST gene recruits or brings in the histone deacetyltransferase to remove those acetyl groups from the histone residues. Long non-coding RNA also recruits methyltransferases. This is really similar to what we talked about in class. We were talking specifically about histone methyltransferases, where these methyl groups are added to the histones themselves. But we'll talk more about this when we get to genetic imprinting and epigenetics, but DNA can be methylated as well. And it has a similar effect as the histone case. So when methyl groups are added to DNA, they form these blockages essentially to proteins that would typically lead to the relaxation of the chromatin structure. So with long non-coding RNA from the XIST gene coding this chromosome here, surrounded in pink, with the addition of methyl groups and the removal of acetyl groups, we have essentially a full compaction of that second X chromosome to the point where it's no longer chromosome shaped essentially. It's essentially a blob. I showed you images of that on the previous slide where you can see this highly compacted heterochromatic version of that chromosome. At this point, all of the genes are effectively silenced, although there are exceptions based on what we currently know about the whole process, but in principle, these genes are silenced, and only gene expression comes from this uncondensed X chromosome. So at this point in time, we have a situation that looks very similar at the chromosome level between males and females, both of them only have one X chromosome that is actively expressing other genes. Neither of these X chromosomes is expressing XIST, because remember, that comes from the chromosome that was condensed. But these other genes that are found on the X chromosome that influence, some of them influence sexually dimorphic characteristics, but genes are also potentially influencing other processes, other phenotypes, and we'll talk about a case of that in just a little bit. Okay, so again, this is the written out form of what I talked through on the previous slide. There are a few remaining questions. One of these has to do with exactly how the cell figures out which one of the two XIST genes to silence. This seems to be a random process, so one or the other X chromosome could be silenced at the blastocyst stage. But how that is perpetuated through mitosis, how that kind of persists through the process of development is still kind of a heavy area of study. And there's also some suggestion recently that at least at some stages, parts of that Barr body might not be entirely silenced. So the process that we talk through here, in terms of what you need to know for course material, you should know this process and its general implications. But as you go forward and you're thinking about implications for future research, for science, for medicine, things like that, keep these things in mind. Okay. So we had a picture of the calico cat on the slide in class, and I mentioned that this was kind of foreshadowing. One of the kind of fun ways that you can see or think about Barr body formation and its impact on phenotypes is through genetic mosaicism. So genetic mosaicism is this effect where you have a mixed phenotype. So this is specifically resulting from differences in genotypes or gene expression levels at different sections of the organism or different patches on the organism. One of the ways that you can get genetic mosaicism is due to monoallelic expression that comes from this random Barr body lionization. So if we think back to these two X chromosomes, you could have a heterozygous individual where one of the X chromosomes is silenced that carries the dominant allele in some cells, and the X chromosome that carries the recessive allele is silenced in other cells in the same individual, resulting in these patchiness of gene expression. So specifically, if thinking about how these two X chromosomes made their way into a female cell in the first place, one of those X chromosomes comes from the paternally derived individual, and one of those X chromosomes is maternally inherited. Okay, so mosaicism in cats is a nice way of illustrating this. So we have a case here where we have a male cat and a female cat. They're both homozygous, but the male cat carries the orange allele and the black female cat carries the black allele. So this color gene is found on the X chromosome. There's a pseudo autosomal region on the Y chromosome that's going to have a similar complement to that gene. So we have both versions available in the X and the Y individual. When these two homozygous parents mate, they can produce monohybrid offspring, thinking about a single gene. So we have our heterozygous male individual and we have a heterozygous female individual. The male case is pretty straightforward. The orange allele is dominant, so its presence on the Y chromosome is going to mask the presence of the recessive black allele on the X chromosome. And in this individual, we have an orange cat. So all of its fur is going to have that orange coloration as a result of the gene activity of the orange allele at this color gene present on the X and Y chromosome. The female case is a little more complicated because of this process of lionization or Barr body formation. So genotypically, the female is also heterozygous. There's a black allele and an orange allele. And if lionization didn't happen, then we would expect this female individual and all female individuals from this mating to be orange. However, we actually get something that often looks like this pretty cat here. So we have a calico female individual. She has orange patches and black patches. If we were to zoom into the nucleus of the orange patch and look at the chromosomes that are present there, we would see the formation of a Barr body as well as a functional gene expression active X chromosome. In these orange patches, we would expect the expression to come from the orange allele, which would mean that this black carrying allele X chromosome has been condensed and turned into a Barr body. So it's completely silenced. Only the orange allele from the X chromosome is expressed. If we go to the black patch and zoom into those nuclei and see what's going on at that level, we would see the opposite case. So in these black cells, only the allele that produces the black pigment is being expressed, which means that the chromosome carrying the orange allele has been inactivated. So no orange pigment is being produced. The only pigment that's being produced is the black, and as a result, we have these black patches of fur. The other coloration you see on this cat, this white big patch, this is actually controlled by a different gene. So this is essentially canceling out the pigmentation activity at we'll call it the orange gene. So this is why you have big patches of white on this particular individual, but you might be familiar with like a tortoiseshell sort of color calico cat where there aren't any white patches and you just see the combination of orange and black. That would be also a case where we have mosaicism due to this variation in Barr body formation. So early on in the blastocyst level development of this particular individual, when we were thinking about that sort of undifferentiated cell stage, part of that blastocyst had this random X inactivation where the black carrying X chromosome was condensed, and another part of that blastocyst had the opposite case happen where that orange carrying allele was silenced. Over time, as cell differentiation continued, as the individual continues to develop, more cells are added to the developing individual through mitosis. This pattern of X inactivation that leads to these black patches and this pattern that leads to orange patches persists and gets bigger and bigger. So, you could imagine if you shrunk this cat down to a ball of cells, there was one cell on this side of the sphere that had this pattern, and there's one cell on this side of the sphere that had this pattern, and they eventually formed these patches as that individual developed. All right. So that was the kind of example that demonstrates a nice visual for genetic mosaicism that relates back to Barr body formation. I just have a few wrap-up slides to kind of recapitulate and put things back into context for what we covered for today. None of this is new information. It's just kind of a way to see these things together. So we talked about eukaryotic DNA organization. We talked about the histone acetyl transferase activity to turn on gene expression, methylation to turn off gene expression, and then a version of these processes as they apply to Barr body formation and X inactivation. All right, and if you're curious about what's coming next, we're going to start talking more about the mechanisms underlying DNA replication. All right, let me know if you have any questions. I'm happy to go over anything. If anything comes up with the last homework questions, let me know, and I will assist as much as I can.